My name is Gülnarə Vagabova, and today we talk about a very interesting topic, enzyme as catalysis. Enzyme is a biological macromolecule produced in the living cells to catalyze the biochemical reaction inside the cell at the body temperature. All biochemical reactions occurring in living organisms depend on catalyzed. It is some metabolic ways which occur in our cells, and you see all these metabolic ways, and all reaction catalyzed by enzymes. Enzymes act in mild conditions such as body temperature and pH. They catalyze the reaction with building and maybe breakdown of materials inside the living organism. Before beginning, I say why enzymes are important. This is metabolic way, and every day upon trillion of chemical reaction occur in our body, and almost all metabolic process in the cells need enzymes, and no enzymes, no reaction. Enzymes are proteins that act upon substrate molecules and decrease the activation energy necessary for a chemical reaction. They are typically highly specific and only bind certain substrate for the certain reactions. Without enzymes, most metabolic reactions would take much longer and would not be fast enough to sustain life. In this lecture, we describe of catalysis. Then we talk about nature, material of catalysis, property of catalysis, their specificity, also classification and regulation, about activation and activation, inhibition, and mechanism of action of enzymes. So if we say about catalyzed, we must talk about other type of catalyzed. It is inorganic catalysts. In this picture, you see major difference between enzymes and inorganic catalysts. First, we say about structure. Enzymes are organic compounds, we say about before, they are proteins. But inorganic catalyzers, they are varies from metal ions, and they have inorganic nature. If we say about place of synthesis, enzymes or organic catalyzed are synthesized by the living cells. They synthesize in our body, but inorganic catalysts are not produced by the living cells. Also, specific property about a specific property of enzymes. Enzymes are usually highly specific in action, but inorganic catalysts are less specific in action. It is difference between these two types of catalyzed. But next, enzymes are sensitive to change in pH and temperature. And sensitive on change, sensitive enzymes have optimum temperature and pH. But inorganic catalysts work well at high temperature. They are not sensitive. Next, we also talk about similarity between these two types of catalysts. Both of them, organic and non-organic enzymes, increase the rate of chemical reaction, but lowering the activation energy. It is both property. Neither change the position of equilibrium of yield of the reaction. Next, enzymes are highly specific, whereas inorganic catalysts are often non-specific and can catalyze several reactions. And we say before about sensitivity of enzymes. And also, I want to say about similarity. Both enzymes and inorganic catalysts are not changed by the reactions. And both of them temporarily bind to the substrate by the reaction. It is similarities and difference between inorganic catalyzed and organic enzymes. So, we continue our lecture and now we talk about structure of enzymes. This is picture which show structure of enzymes. Enzymes are proteins, and it is demonstrated its composition. All enzymes consist of amino acids. That's why they have all property of proteins. We say about before, when we study chemistry of proteins, and same as a protein, they have divided for two groups: simple proteins, or we say simple enzymes, which consist of only amino acids, and complex. Complex enzymes consist of protein part, sequence of amino acid, and non-protein part. They called complex enzymes. Complex enzyme, as you see in this picture, divided for two groups. It is protein part and non-protein part. Protein part called apoenzyme. Non-protein part coenzyme. Both of these parts, generally complex protein, name will be holoenzyme. The holoenzyme divided for two parts, apoenzyme and coenzyme. As you see in this picture, coenzyme maybe have different name. It's maybe prosthetic groups and it's maybe cofactors. It's depend from non-protein part, how this non-protein part bind to apoenzyme. Coenzyme may be bind weakly, maybe bind strongly. If non-protein part binding strongly and formed strong covalent bond, this part called prosthetic groups. But cofactors, some enzyme associated with non-protein, it is small units. If enzymes have small units. It's maybe ions, it's maybe metals, such as zinc, iron, copper, or other molecule. These small compounds called cofactors. Coenzymes, if we say about nature, coenzymes, they may be vitamins nature and non-vitamins nature. And the next picture illustrated composition of enzymes. What here you see apoenzyme and cofactor, it's maybe coenzyme, and form holoenzyme. Here protein and non-protein part generally will be holoenzyme. And it is enzyme in this picture, illustrated enzyme, it is inactive protein. If enzymes are complex, this enzyme need non-protein part or cofactor, we say. For example, some metals. Without this cofactor, this enzyme will be inactive. Inactive protein connecting with coenzyme or cofactor, and after this connecting, formate active enzyme. Also, after connecting, this enzyme can used in reactions. Next picture demonstrated some coenzymes and cofactors. 35% enzymes used metals, and here you see metals and names of enzymes which need for its action metals. For example, zinc, carboanhydrase, carboxypeptidase, and other. This is names. And before I say about coenzyme, some of coenzyme, many coenzymes have vitamin nature. It is water-soluble vitamins. All water-soluble vitamins have coenzyme form in our body, and these coenzymes is part of enzymes. When we will study metabolism, we will talk about it. And now I show some enzymes which have vitamins. For example, vitamin B1, it is thiamine. Coenzyme form is thiamine pyrophosphate. It is coenzyme part of pyruvate dehydrogenase complex. It is general pathway in our body used in this reaction. Flavin B2 vitamin and nicotinamine B3. This is vitamins have coenzyme form flavin adenine nucleotide and nicotinamide, short name it NAD. They used they coenzymes of oxidoreductases is also main coenzymes which used in respiratory of our cells. Next, vitamin B6, pyridoxal. It is coenzyme form of pyridoxine vitamin B6, pyridoxal phosphate. It's used in transamination reactions, transfer amino groups, and also you see here enzyme glycogen phosphorylase. It is metabolism of glycogen. Also biotin, coenzyme A, and vitamin B12 also used in the main metabolic ways in our body. More information about this coenzyme form we will study in the next lecture in the when we will describe vitamins. In the next picture, we see a graph. In this graph, we comparison activation energy of uncatalyzed reaction, which show in this graph in violet curve, and green curve show activation energy of catalyzed reaction. Let's consider activation reaction. Before, at the beginning of this lecture, I say that enzyme increase the rate of reaction by lowering activation of energy. What is it, activation energy? In biochemical processes, molecules need energy in order to start a reaction. For example, molecule need have some kinetic energy to collide with other molecule to start a reaction. If the collision don't happen often or don't have enough kinetic energy, reaction will not take place. The energy required to start a reaction is called the activation energy. The lower the activation energy, that the faster a reaction happens. What we see in this graph, reactant and product have specific energies. In order to transform the reaction into product, the reactant would have to go through a transition state, intermediate state, which is usually higher in energy. To get to this transition state, the system requires the activation energy. Finally, the product, which we have at the end, reduce the energy to arrive to the final product. Reactants have higher energy, it is reactant. Reactant have higher energy than product and product. The energy of the reactant increase at the end, first increase energy of reactant, first increase, then decrease to the final product energy. The highest point in this curve, the highest point in this curve, represent the energy of the intermediate state in these reactions. Intermediate state in this reaction, enzyme lower the activation energy of a given reaction shown by the green curve. And it is proof that enzymes decreased the activation energy. So now we talk about main question of this lecture. It is mechanism of action, enzymes. To catalyze a reaction, an enzyme will bind two or more reactant molecules. The molecules are the enzyme's substrate. In some reactions, maybe one substrate is broken down into multiple products. In other, two substrates come together to create one larger molecules to swap pieces. In this picture, you see one of the million reactions in our body. This is a reaction of carbohydrate metabolism, and it is a reaction of synthesized active form of glucose from glucose in our body. Synthesis glucose 6-phosphate. It is active form of glucose. This enzyme catalyzes this reaction enzyme hexokinase. In this picture, you see this enzyme hexokinase, and as you see, enzymes, we say before about it, enzymes are big compound because they consist of more amino acids. But substrates are smalls. That's why when reaction begin and enzyme substrate are connected, this small compound substrate connecting only on the specific part, specific site of these enzymes. And these sites called active site, this part of enzyme called active site. And at the end form product of this reaction. What is it active site? The part of enzyme where the substrate binds is called the active site. It's part where catalytic action happens. Proteins are made of units called amino acids, and the enzymes that are proteins. The active site gets its properties from the amino acids it's built out of. These amino acids may have side chains that are large or small, acidic or basic, hydrophilic or hydrophobic. It is a special part which binds the substrate and convert it into the product to the reaction. The substrate is bound in the active site by the multiply weak forces. It is maybe electrostatic interaction, hydrogen bonds, van der Waals bond, hydrophobic interaction, and maybe some cases by reversible covalent bonds. The active site contains amino acid side chain that create a three-dimensional surface complementary to this special substrate. The active site binds the substrate, forming an enzyme substrate complex. And this site contained one or more binding sites that orient the substrate in the correct configuration, and also a catalytic site which in the part of the molecule that lower activation energy. The remainder of an enzyme structure acts primarily to present the active site to the substrate in the best way. Also, maybe some enzymes have allosteric site. In this part, bind activators and inhibitors, and they change conformation of enzymes and change activity of this enzyme. These compounds regulated activity of enzymes. We consider steps of enzymatic catalysis, and now you see in this picture substrates and enzymes and enzyme. Two different substrates work together in the active site, bind to enzyme, and form the enzyme substrate complex. We know two explanations of how enzymes interact with substrates are the log and K model. This model proposed by Emil Fischer. It is a matching between an enzyme's active site and a substrate isn't just like two puzzle pieces fitting together. It is a model called lock and key model. Instead, an enzyme second model, enzyme change shape slightly when it binds its substrate, resulting in an even tighter fit. This adjustment of the enzyme and substrate conformation called induced fit model. The induced fit model proposes enzyme molecules can change their shape depending on the interaction with the substrate. In this model, the enzyme and sometimes the substrate change shape as they interact until the active site is fully bound. It is to model how enzyme and substrate connect, bind. After this binding, enzyme and substrate, as we say, and the substrate connected in the active site, will be second step. Second step, first step, enzyme and substrate binding and formate enzyme substrate complex. After this step will be slowly, and in this step we have many reactions. To be able to make a bond, maybe break a bond, have many reactions of chemical transformation of this substrate. This steps called covalent catalysis, and covalent catalysis occur when one or multiple amino acids in the active site form a covalent bond with the substrate. This reaction usually takes the form an intermediate through attack of the catalyst results with the help stabilize later transition state. In the next step, general acid-base catalysis takes place when a molecule other than water acts as a proton donor or acceptor. Water can be one of the proton donors or acceptors in the reaction, but it's not only one. A common example of this involves water entering the active site, donate or receive of proton, after a substrate has already bound in order to form better nucleophiles that can form and break bond either. If we have metal in enzyme, metal ion catalysis involves the precipitation of a metal ions at the active site of the enzyme, which can help to may attack a better nucleophile as well as stabilize any negative charge in the active site. In this step, after this many reaction, step will be chemical step. First step, we must call it physical step because also change configuration of substrate and enzymes. In this step, it is chemical step. In this step occur many reactions in this complex and begin formation of product. After this step, release of product and form product which we need for next reaction, and free enzyme. Enzymes are typically highly specific and only bind certain substrates for certain reaction. Specificity, it is selectivity of enzyme to these substrates, is the ability of the enzyme to choose an exact substrate from a group of similar chemical molecules. The specificity is actually a molecular recognition mechanism, and it operates through the structural and conformational complementarity between enzymes and substrate. Enzymes show different degrees of specificity towards the substrate. You see in this picture types of specificity of enzymes. We divide it generally for three groups: absolute, relative, and stereospecificity. First, absolute specificity. It is also, we say, substrate specificity, also we call it absolute. It seems here the specificity is very high. Enzyme showing substrate specificity are specific only to one substrate and one reaction. It shows that this enzyme in our body, in our cells, catalyze only one reaction and have very high specificity for only one substrate. For example, lactase can only hydrolyze glycoside bonds of lactose, for example. Maltase to maltose, for example, and urease decompose only urea, or arginase split only arginine. Next type is relative specificity. Also, we say it's call it bond specificity. Enzymes showing bond specificity are specific to substrate having similar bond on similar structure. These mean that they are specific only to certain types of bonds, as peptide bonds, maybe glycoside bonds, maybe ester bonds, and other. If we say examples, it's maybe enzymes of gastrointestinal tract. Maybe we can say alpha amylase. Alpha amylase, it is enzymes which hydrolyze alpha-1,4 glycoside bonds. It's in glycogen and in the starch. Or, as you see here, pepsin. Pepsin, it's peptidase of our which synthesis in our stomach. It's only hydrolyzed peptide bonds. And split different proteins, not only one protein, different proteins. And also maybe lipase. Lipase also hydrolyze ester bonds. Next type, it is stereospecificity. Also other name, optical specificity. Here the enzyme is specific not only to substrate, but also to its optical configuration. Optical specificity of enzymes is considered as the highest specificity shown by any class of enzyme in the living world. It may be L and D trans and cis form of different compounds. For example, L amino acid oxidases act only L amino acids, not act to D amino acids. But D amino acids oxidase only act for D amino oxidases. Other main property of enzymes, it is effect of temperature, pH, and substrate concentration. It's all factors. From these factors depend activity of enzymes. First graph, this first graph show how activity of enzyme depend from temperature. Enzymes function most efficiently within the physiological temperature range. Since enzymes are protein molecules, they can be destroyed by high temperature. But changing the temperature can also stimulate enzymatic activity. You see it in the graph. When we increase temperature, enzymatic activity also will be increased. Increasing temperature has two effects on an enzyme. First, the velocity of the reaction increases somewhat because the rate of the chemical reaction tends to increase with the temperature. But when temperature till 35-40, it is maximum temperature. After this, enzyme is denaturated. Increasing temperature, this increases the metabolic rate only within a limited range. It's our body temperature. If the temperature becomes too high, enzyme denaturate and lose its activity. Enzyme destroy life, lose activity, and denaturated and lose its function. It's unreversible process. Low temperature also change the shape of enzymes. Low temperature. With enzymes that are cold sensitive, the change causes loss of activity. Enzyme in the negative temperature loses activity, but when we will continue and will increase the temperature, the activity of enzyme will recover, and its function of enzymes used in the medicine for long storage organ and tissue. Other property of enzyme, it is this shown in this graph, it is effect of pH. Enzymes are affected by change pH. The most favorable pH value, the point where the enzyme is most active, is known as the optimum pH. Well, the optimum pH in our body, physiological pH from 6 to 8. It is optimum pH in our body except stomach, as you know. This property illustrated in this graph. Extremely high or low pH values generally result in complete loss of activity for most enzymes. pH is also a factor in stability of enzymes. As with the activity for each enzyme, there is also a region of pH optimal stability. And in this graph shows how enzyme activity depends on substrate concentration. When substrate concentration increased, activity and rate of the reaction also will be increased till to saturation point. When will be saturation point? After this, rate of reaction unchanged. In the next table, we see the optimum pH value some enzymes we see in this table. For example, what we see from 6 to 8, it is some enzyme of our gastrointestinal tract, sucrase. It is absolute specificity. We say before about specificity, its enzyme destroys saccharose to fructose and glucose. Amylase also in pancreas and oral cavity, alpha amylase hydrolyze glycoside bonds. And trypsin lipase, its enzyme of gastrointestinal tract, except urease, it's in liver. Also pepsin synthesized in our stomach, and as you know, in our stomach secreted HCl, and that's why in our stomach pH strong acid, and this is pH optimum for pepsin. Now we talk about regulation of enzymatic activity and about activators and inhibitors of enzymes. Enzymes can be regulated by other molecules that either increase or reduce the activity. Molecules that increase the activity of enzymes are called activators. Activators is inorganic substance that tend to increase the activity of enzymes, but inhibitors, while molecules that decrease the activity of an enzyme called inhibitors. That's inactivated it. Inhibitors may be competitive and incompetitive. And now we describe its action. First, we say about activators. Enzyme activators are chemical compounds that increase a velocity of enzymatic reaction. Their action are opposite to the effect of enzyme inhibitors. Among activators, we can find ions, small organic molecules, as well as peptides, proteins, and lipids. There are many enzymes that are specifically and directly activated by small inorganic molecules, mainly by cations. Small molecules can bind to only with the enzyme, but also with the substrate, increasing its affinity to the enzyme, and that activate this enzyme. What it is show, it's demonstrated its picture. Special groups of activators can produce activation of target enzymes only after the formation of complex with another molecules, that after connecting activation, this complex is turned bind to the enzyme and increase the velocity of this activity. Next, about inhibition. It is, you see, a type of inhibitors. Enzyme inhibition maybe have two types, maybe reversible and irreversible inhibition. And reversible inhibition divided for some groups: competitive, uncompetitive, and non-competitive inhibition. An enzyme inhibitor is a molecule that binds to an enzyme and decrease in activity. But it is competitive inhibition. It's photo demonstrated mechanism of competitive inhibition. Some molecule very similar, the substrate, enzyme, substrate, and inhibitor. Its structure, form, it is very similar to the substrate. For some molecules very similar to the substrate. For an enzyme may be bound to the active site, and this site inactivated. Well, as you see here, it is enzyme and connecting to active site inhibitor, and enzyme this substrate cannot bind to enzyme, and reaction will stopped. And as you saw, no reaction. It's blocked the active site, block active site. These inhibitors are of competitive nature because the inhibitor molecule binds to active site. For example, the nerve gas disopropyl fluorophosphate, it is example, react with the serine at the active site of acetylcholine esterase to form a covalent bond. The nerve gas molecule involved with the bond formation prevent the active site from the binding the substrate. Acetylcholine, it is substrate and blocking catalysis and nerve action. Other example, iodacetic acid similarly block a K enzyme in muscle action by forming complex with amino acid cysteine, which is bound, which is found at the enzyme active site. Well, this also picture for which show these competitive inhibitors. Competitive with substrate for binding to active site, but once bound substrate cannot transform it into product by enzyme. Only inhibitor connecting with bind to the enzyme, but competitive inhibitor can be reversible by simply increasing concentration of specific substrate of this enzyme. In non-competitive inhibition, inhibitor modify amino acids other than those at active site, resulting in the loss enzymatic activity. It is compare competitive and non-competitive inhibitors, and what here you see. The inhibitor causes changes in the shape of the active site. However, it can be modified without affecting the structure of the active site. In these cases, enzymatic action is not affected. In competitive inhibitor bind only the active site. Uncompetitive inhibitors connecting non-active site in other part of enzyme and decreasing enzymatic activity. These inhibitors, we say non-competitive because they inhibitors and substrate can both be bound at the same time. This inhibitor attached at another site and blocks the enzyme from doing its job. Other type of regulation, it is allosteric regulation. Inhibitors and activators that bind to enzyme not in the active site, but in the special center. But not active site, it's special center. Located far enough from it have name allosteric modulators. Their binding to allosteric sites induced the change of the enzyme conformation that affect both the structure to active site and enzyme conformational mobility, leading to the decrease or increase of enzymatic activity. Just an enzyme active site is specific in relation to substrate, the allosteric site is specific to its modulators. Many metabolic pathways are regulated through the activation of allosteric modulators. Enzymes in metabolic pathway work sequentially at its such pathway. A product of one reaction becomes substrate for the next one. The rate of whole pathway is limited by the rate of the lowest reaction. Allosteric regulators often are a final product of whole metabolic pathway that activated enzymes catalyzing a limited step of the whole pathway. Enzymes in a metabolic pathway can be inhibited or activated by downstream products. This regulation represents negative and positive feedback that slow metabolic pathway when the final product is produces a large amount, or accelerate in when a final product is presented in low concentration. Allosteric modulators are important participants of such negative and positive feedback in metabolic pathways or between the making metabolism self-controlled. Any of a group of proteins that display no catalytic activity but are transformed within an organism into enzymes, especially those that catalyze reaction involving the breakdown of proteins. Its compounds, its enzymes called proenzymes or zymogen. They synthesis in non-active form and after reactions of proteolysis formed active form of this enzyme. For example, trypsinogen and chymotrypsinogen, these zymogens secreted by the pancreas and are activated in the intestinal tract to trypsin and chymotrypsin. Activation is affected by the cleavage of one or more peptide bonds of the zymogen molecules and may be catalyzed by a separate enzyme enterokinase, convert to trypsinogen to trypsin. Zymogenic cells synthesize and store zymogen in inactive form. Next. Other form of enzyme, it's called isoenzymes. Isoenzymes are different form of an enzymes which catalyze the same reaction but which exhibit different physical or kinetic properties such as isoelectric point, pH optimum, substrate affinity or effect of inhibitors. Different isoenzymes form of enzyme are usually derived from different genes and often occur in different tissue of the body. An example in this picture, you see enzyme lactate dehydrogenases. Its short name LDH. LDH. This enzyme catalyzes in our body the reversible reaction, conversion of pyruvate into lactate, the presence of the coenzyme NADH. LDH, not enzyme, it is tetramer of two different types of subunits, H and M, which have small difference in amino acid sequence. The two subunits can combine randomly with each other and forming 5 isomers. It will be LDH1, which consists 4 H subunits, and it is in which organ occur this form. LDH2 consists of 3 H and 1 M form. Next isoform will be LDH3, which have 2 M and 2 H form. LDH, lactate dehydrogenase form, isoform number 4, consists of 3M and 1 H form. And at the end, LDH5, which consists of 4 M subunits. It is different form of different isoform from one enzyme, lactate dehydrogenase. This enzyme catalyzed one reaction, but amount in organs and tissue will be different. And each organ and tissue have special form of this enzyme, maybe LDH1, LDH2, 3, and 4. And amount of these isoenzymes in the blood used in diagnostic of disease. Now we talk about classification of enzymes. According to the International Union of Biochemists, enzymes are divided into 6 functional classes and are classified based on the type of the reaction which they are used to catalyze. In this table, you see 6 classes of enzymes. First class, it is oxidoreductases. Oxidoreductases, it's enzyme which catalyzes the oxidation and reduction reactions, where the electrons and protons tend to travel from one form of molecule to the other, and include dehydrogenase, oxidase, peroxidase, monoxygenase, included this enzyme. It is an example of reaction in this class. Enzyme of lactate dehydrogenase, we say about it in the isoenzymes, and we describe isoform of lactate dehydrogenase. It is this reaction, lactate transferred to pyruvate, transform it to pyruvic acid, and this hydrogen, lactate dehydrogenase, this hydrogen connecting to NADH and form NADH, enzyme lactate dehydrogenase. Second classes, it will be transferases. Transferases help in the transportation of the functional group among acceptors and donor molecules. Transfer some groups from one compound to other. It is names of this enzyme depend from which group it's enzyme transfer. For example, aminotransferase, glycosyltransferase, or phosphotransferases. It is next class, transferases, and example reactions, alanine transaminase. It is enzyme transfer amino group from one compound, amino acid alanine, to keto acid, alpha ketoglutaric acid, and formed pyruvic acid, new keto group, and amino group. These two compounds change amino and keto group. Next class, hydrolases. Hydrolases are enzymes, are catalysts, hydrolytic enzymes, which catalyze the hydrolysis reaction by adding water to cleave the bond and hydrolyze it. There are also names. It's schematic. We see mechanism of this reaction and example names of these enzymes. It is reaction which catalyzes hydrolases. Hydrolase reaction where used water. Next class will be liases, or other name synthase. We used liase in these classes, enzyme of these classes. This enzyme catalyze reaction where non-hydrolytic removal of group or addition of a group to a substrate. And in this reaction, maybe destroy these bonds, and maybe synthesis new double bonds. It is lyases. And now I show you this example. Pyruvate decarboxylases. And here formed dioxide, carbon dioxide. Remove this group and form carbon dioxide. And here synthesis, double bonds with CO2 product. Fifth class, it is isomerases. Isomerases catalyze the structural shifts present in the molecule, this causing the change in the shape of the molecule, catalyze the formation of an isomers of compounds, change of the molecule form of the substrate. What it is isomerase, reaction of isomerization, change the molecular form, and enzymes, maybe epimerase, isomerases. Also, I show you reaction example. What catalyze it isomerases alanine racemase from L form synthesis D alanine. It is a reaction of isomerization. And at the end, sixth class, it's ligases or synthetases. Its group are known. Its group are synthetases, and these enzymes catalyze the association of two molecules. And in this reaction, used ATP, use energy, glutamate and ammonia connecting. We use energy ATP and form L-glutamine. It is glutamate amino acid from glutamate form glutamine. It is a reaction of synthesis and here or synthetases, and now it is mechanism of this reaction. All enzymes have specific code for number. It is code have all enzymes because in our body we have many enzymes, and in the and we used this code. Let's show this code. First number represented the class. We say about 6 classes of these enzymes. Second digit stands for subclass. Third, subclass. And fourth, serial number of the particular enzyme in the list. Now we talk about enzymes in clinical medicine. The study of enzymes is termed enzymology, and the principles of enzymology outlined previously are applied clinically in 3 ways. First way, it is enzyme pathology, when enzymes used for diagnosis and prognosis of disease. Also, enzyme diagnostic, enzymes as a diagnostic reagent, and enzyme therapy when some enzymes we used as a therapeutic agent. Now you see main serum enzymes which used for diagnostics. The measurement of the serum levels of numerous enzymes has been shown to be of diagnostic significance. This is because the presence of this enzyme in the serum indicates that tissue or cellular damages has occurred, resulting in the release of the intercellular compounds into the blood. Enzymatic diagnosis is a method to diagnose disease by measuring the content and change of certain substance in the body, or through the change of the original enzyme activity in the body. It is evident that enzymatic diagnostic method based on two aspects. First, when changes in the original enzyme activity in the body and change of certain substance in body fluids, which is detected by enzymes. For example, phosphatase is a hydrolase that catalyzes the hydrolysis of phosphate monoesters into inorganic phosphate under acidic conditions. Serum acid phosphatase activity will be elevated in the body of patient with prostate cancer or hyperparathyroidism. Another example, for example, glucose oxidase can be used to detect glucose content in diabetes diagnosis. Also, lactate dehydrogenase, we say about this lactate dehydrogenase, so this enzyme, when we talk about as an enzyme, and creatine kinase, this is creatine kinase. Also, it's called creatine phosphokinase, are commonly measured in the diagnosis of cardiac infarct. Gamma glutamyl transferase, this is or GGT with short name, also called gamma glutamyl transpeptidase, is involved in glutathione metabolism and also in amino acid transport across membranes. GGT levels in blood is of gamma glutamyl transpeptidase, or levels in the blood, is diagnostic for disease of liver and the biliary systems, as well as disease of the pancreas. Also, the measurement of lactate dehydrogenase is especially diagnostic for lactate dehydrogenase. It's especially diagnostic for myocardial infarction because this enzyme exists in 5 closely related by slightly different forms, or we say about isoenzymes. These 5 isoforms are generated by a combination of 2 different subunits encoded by 2 different genes. The subunits are identified as M form for muscle and H form for heart specific. 5 types of LDH used in diagnosis. In a myocardial infarct, the serum level of LDH rises within from 24 to 48 hours, reaching peak by 3 to 3 days and return to normal in 5-10 days. Especially diagnostic is a comparison of the LDH1 and to LDH2 ratio. Normally, this ratio is less than 1. The typical liver enzymes measured are aspartate transaminase ALT and alanine aminotransferase or alanine transaminase ALT, or the short name. When assaying for both alanine aminotransferases and aspartate aminotransferases, the ratio of the level of these two enzymes can also be diagnostic. Normally, in liver disease or damage that is not of viral origin, the ratio ALT to AST is less than 1. However, with viral hepatitis, the ALT to AST ratio will be greater than 1. Measurement of AST is useful not only for liver involvement, but also for heart disease or damages. The level of AST elevation in the serum is directly proportional to the number of cells involved as well as on the time following injury that the AST assay was performed. Next wave of enzymology, it is enzyme therapy. And I talk about some example when we used enzymes in medicine. Enzyme therapy is a plan of dietary supplements of plant and animals enzyme used to in the digestive process and improve the body's ability to maintain balanced metabolism. In traditional medicine, enzyme supplements are often prescribed for patients suffering from disorders that affect the digestive process. It's main. Then, for example, plasminogen. For example, plasminogen is converted to plasmin, which is a fibrinolic compound. Next, collagenase used in the skin ulcers causes collagen hydrolysis, break up and remove dead skin and tissue. Next, prolactoenzyme used in lactose intolerances. About 75% of the world's population is intolerant to lactose in adulthood. It occurred due to lack of lactase in digestive system. Also, streptokinase enzyme in myocardial infection used as clot blaster to dissolve clot in the arteries of heart walls.